Hello, and a very warm welcome to everyone joining us for today's Select Science webinar entitled Maximize the Impact of Visualization-Driven Omics Data Analysis. My name is Matt McArdle, and I'll be moderating today's presentation. I'm delighted to introduce today's speaker, Carl-Johan Iversen, who is the Qlucore president. In the webinar today, he's going to be talking about the benefits of visualization-driven data analysis, especially omics data analysis, how fast and highly interactive software drives improved results, how visualization can be used to easily and efficiently share results and ideas. After the webinar presentation, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A session at any time during the webinar. You can submit your question at the bottom of your screen. We're also going to be running a few audience polls today during the webinar. And to get us started, we'd just like to ask a couple of questions to the audience. These should be popping up on your screen now. And our first one is, how often do you use bioinformatics software? Do you use it daily, weekly, or monthly? Just give the audience a couple of seconds to answer that. And our second question we're going to ask today is, what type of data do you work with the most? Is it gene expression arrays, gene expression for RNA sequencing, single-cell RNA sequencing, proteomics, or DNA methylation? Thank you. So without further delay, I'm delighted to hand over to Carl, and I'd like to thank him for presenting to us today. Please go ahead, Carl.
Thank you, Matt. So a warm welcome to all of you. Visualization-driven data analysis have many benefits. The most obvious is that you as a user actually sees and can control the analysis through all steps. Today's webinar will be very visual, and I will demonstrate live using the Qlucore Omics Explorer software. I will demonstrate how visualization will add significant value to your analysis. Before we start, a warm welcome to you all. The objective is to show how visualization-driven analysis generates improved results and how visualization makes it easier to work in teams. But before we start with the actual visualization and demonstrations, I would like to tell you a little bit about Qlucore. We have customers in 25 countries. We were founded in 2007 and we originate from Lund University in Sweden. 3 out of the 5 largest pharma companies are our customers, and we do new product releases every 6 months, and over time we have made 9 major releases. We usually say that all well-known universities are our customers, and one way to check that for you is to go to Qlucore.com, and where we have a list of all publications in peer-reviewed journals. Currently, there are a little bit more than 600 of these publications that we have found. And in addition to providing information about who actually is using Qlucore, it also gives you it's a great resource for finding out what they actually have done using the program. As you can imagine, users work with many types of data. It spans from multivariate data from a broad range of experiments to NGS data. The lists here on the slide really gives you an overview of the opportunities, but it's not limited to these. In addition to the experimental data that you have measured, annotations and clinical information can easily be imported and used. And you will see examples on that later on. users and organizations we meet highlights different challenges, but for a vast majority, the following four ones are highly relevant. So most organizations are struggling with finding new things in data. They say that they are lacking bioinformaticians or statisticians in the organizations. Overall, the new technology means that size and complexity distances biologists from the experiment. And people also tend to think that existing software is really too complicated to use. Qlucore's idea on how to solve these challenges is to work with visualization-driven analysis. In short, the idea is to skip the step where you work with data in tables and move directly to visualization. Then you as a user can apply filters, do selections, and use statistics at the same time as data is visualized. The visualization-driven analysis enables the user to inspect data during all analysis phases, see changes when filters are applied, continuously control the analysis process. I will now illustrate this in the first out of 3 demonstration sessions, where I will run a live demonstration with Qlucore Omics Explorer. And demonstration 1 will show you how you quickly and easy can do an initial inspection of a data set and how that inspection can be verified towards known annotations or clinical information. I will show you multiple visualizations and filters, and I will also show an example on how visualization can safeguard you from missing important information. So I'm going to switch to Qlucore Omics Explorer, which I already have started here. I have also loaded a data set. It is a data set of children who have leukemia. There are 132 samples measured. And you can see that down to the left here. It's a gene expression data set, so we have 13,000 variables or gene expression levels that are measured. When you start Qlucore Omics Explorer, you will always be presented with a plot. And in this case, it's a PCA plot, principal component analysis. And the nice thing, or one of the nice things with PCA, is that it has a very natural interpretation. So in this case, 2 spheres that are close to each other have a very similar expression pattern, and 2 spheres that are far from each other are different. So that's the way to interpret the PCA plot. And you can see we have 3 axes, so it's a 3-dimensional PCA plot with the first 3 principal components being used and data visualized with that. Before we actually go into the exploration and inspection of this data set, I would like to give you a quick tour of the user interface so that it will be easier for you to follow what I do at later stages. But I will also say already now that I'm not going to give you all the details and all the functionality in the program. So this will just be snapshots and I'm focused on some areas of the program. But up to the left here, you have the toolbox where you control the pointer. So now it's in move mode. If I would select label mode, I can label a sample. And I can clear it. I can also label by pressing in tables like this. There are many more options here, and I will use some of them as we do the demonstration here. Below the toolbox, you have 4 tabs. Samples include sample annotations, so clinical information. The variables include variable lists, which you can either create in the program or import. The log functionality manages information about what we have done to the data set, what type of filters and tools we have applied to it. And it also gives you possibility to log exactly what you have done. I will show that later on. The NGS tab handles the NGS module, which we will not touch at all today. Up here, you find a number of other tabs. We will focus on method and view today. And in the method tab, you select the plot type. Now it's PCA and it's in samples. If you, for instance, select a heat map will be generated on the data, and quickly you can go back. The statistics dialog is where you control the majority of the statistical filters and how they are applied to the data set. So, as you can I still have a label here. We should clear that. As you can expect, leukemia should have subgroups. You should be able to see subgroups in your data set, which you currently are not in the PCA plot. And the reason for that is that there are also very many variables that has nothing to do with leukemia, so they contribute noise. So the first thing we need to do is to reduce noise levels, and a filter that is excellent for that is to filter by variance. So we will remove variables from the data set that has very little variance. And you do that by just moving this slider slowly, and at some point the plot starts to update and you can start to see things. And this is really what we mean with visualization driven analysis. You change a filter and you see the results immediately. That gives you full control and full freedom, and the speed that you get the update is more less instantly, so you can test many different settings in a short time. We can directly see in the plot here that there seems to be structure in this data set as we expected. Then the question comes, okay, is this a good representation that we have right now? Should we filter like this or filter in a different way? To help you with that, we have this metric here called projection score. You can read more about that in white papers from us, but it helps you to find a representation of your data set that shows as much information as possible. So what we will do is we try to maximize that. So somewhere here seems to be a good plot and good settings. And you can clearly see that there is one group up here and maybe more groups down here. So let's store this information now that we can see multiple groups. And the way to do that is to have an annotation. So I create a new annotation here. We can give it a value, webinar, and we can make sure that there are subgroups in that annotation. And then to collect the information that this is a separate group, the way to do that is to select color, not necessary, but we'll make it easier. Then select a group without members, select the annotation tool, and just circle those samples. So now we have divided the data set in two groups. 118 samples or patients is in the blue group, and 14 samples are in the yellow group. We still don't know what the biological meaning of these groups are. We are just inspecting the data using tools to find subgroups and structure. As you saw from how I used the pointer here, you really achieve this and create the annotation and the coloring and selections with just a mouse, and it's really convenient. So to inspect the blue part here, the blue samples in more detail, we can remove the yellow group, and we felt that before that we saw that were groups down here, but we can, to enhance that, we can use the nearest neighbors. So then we connect the nearest neighbors, and we can more or less directly identify more groups here. So let's do that very quickly. Move that a little bit. White is not optimal, so let's change. And that is how easy you change color. And maybe there are more. So these here are quite close. Should you include those 4 or not? Should you include those or not? And those are questions that you could consider. Same thing here. If you do like that, where do they actually belong? They start to be closer to that one. So we will not do that now, but we can remember that for later stages that there potentially could be refinement. What I would like to show you now is that there are more options in the program to find structure than the PCA plot. We could have continued to work with the PCA plot, but just for the sake of it, show you another option called isomap, where you look for have a different way of calculating distances between samples. So you go through a network instead, which can enhance plots. As you see, we pick out the same groups we picked out here, and the interpretation of this plot is the same. So samples that are close to each other in the plot are similar. You can also see that here there is some not so clear. That was clearer in the PCA plot. So the 2 types can really help you. But here is an arm of samples that we very clearly can add. So we annotate that. And remember, we are generating a hypothesis. So at this point, it doesn't matter if we take one sample more or less in the groups. We can refine that at later stages. So this is how you can use isomap, one option. We can clear that, and we go back to the PCA plot. And you see, maybe then the PCA plot, maybe those should be kept as blue and so on, but it starts to indicate that. More options is to go for a T-SNP plot. And you do that by as easy as this. And then we would like to color that. And as you see, there are some of the samples that probably very clearly here shouldn't be grouped as they were. We can correct that. So we make that one blue and we make those one blue. But we can also see that there are potential of more subgroups. And so let's just show you how you can colors here again and annotate, and we can annotate those. So you work with the program in the same way regardless of what plot you are using, and so it's easy to learn. And you see those 4 here, that was the one that we already saw in the PCA plot, and they are also here, so that also gives you confidence that different types of methods gives you similar structures in your data set. That's, of course, very convincing. And we go back to the PCA plot. And it makes some sense even here, which is good. So let's see, have we found something that is biological relevant or not? Before we go into that analysis, I will I will create a log point. I will tell you more about that later. when we use it, but I am right now storing a snapshot of the program, so I can keep track of what I have done, and I can go back to this point later, which I will do. We should give it a name, initial inspection, we can say. Let's now move on to understanding if this information we have collected, the new annotation we created, actually have some relevance to from a biological point of view. What I'm doing now is that I am creating a so-called synchronized plot. So it's two plots that share the same data and the same statistical filters and selections. But it gives me the freedom to color according to different annotations. I actually also have the freedom to have a different plot type. So I can of course have a t-SNE plot, and these are both synchronized. But it makes more sense to have the PCA plot, and that also shows you one important thing, how easy it is to go back, and it remembers my coloring and so on. I will close that down so it's easy to see. Okay, we identified that yellow group, and looking at the other synchronized plot here, which I now have colored according to the annotation called leukemia subtype, which is a classification that was made by the physicians who did the original study. I can see, or we can see, that the yellow samples here is equal to the orange ones here. That's clear. And they are the TLLs. So clearly a biological relevance finding that we found. And if we continue to inspect our results, we can see that we found that purple group, which equals the red group, the E2A PBX1s. We also found that green group, which equals the blue one, the TLAML1s. And we found a lot of these white samples here, which equals the hyperdiploid and so on. And if we would have continued, we would have found even more subgroups. We also found that blue one here, which doesn't necessarily seem to have be visible here in the other group. But if I change annotation here to someone that is called novel group, which was the finding that I did in this study, that I identified a new group that was a subgroup in other. Actually, we can see that with our visual inspection, that was exactly what we identified. So visual inspection gives you real biological relevant information. So you can also complement your visual driven analysis with other tools. So for instance, we have clustering based on k-means plus algorithm. So it's an unsupervised model. And if we would have had more time, I could have showed you how to do that. And then you would have gotten another view of how the clusters in the data should look. And it would maybe given you even more knowledge about your data set. But we will not do that today. So instead, we move back to the slides here. And to summarize our first demonstration, we found structure very quickly. We had full control of the data set. We used multiple options to look into and dive into the data. And all we needed was checkboxes, move of sliders, and simple controls run with the mouse. Very, very, very easy to learn. Now we will move on and demonstrate how the user and the organization can benefit on a higher level from the visualization-driven analysis, since it enables the user to test many more ideas, do both easy and advanced analysis without being an analysis expert, utilize domain knowledge for deeper insights, such as the domain knowledge that a biologist or medical doctor or chemist has into their specific research area. And importantly too, when a biologist or non-analyst is starting with their own experiment data, they can also then ask better questions to the bioinformaticians, the statisticians, which saves time and improves the overall results. So the second demonstration will show you how the visualization gives deep insights into data and why it can be very useful to combine visualization with statistical tests. You will also have seen an example on how quickly you can verify results in a second data set and how easy it is to communicate results for using, for instance, a PCA plot. So let's go back, and then I will close that and make sure I don't have any filtering on. Then I will do something that is not the preferred workflow, but to give you an example on how important it is actually to not use a table and not avoid and make sure that you always look at data at the same time as you work with it. So these steps here is more of the traditional way of applying statistical filters to data sets. You have the tables, and then you apply statistical tests, and then you look at the results. So let's do that, and I will come back later with more information on how you should do it instead. In the statistics dialog, we select a two-group comparison because we're going to do a t-test. We are going to do it to find discriminating variables for a group called TL-AML1. And there are different options to select your cutoffs. Here I select the false discovery rate of 0.05. And when I press that, the filtering is done. And if we look down to the left here, you can see that with that setting, we find 1,700 variables, in this case genes, that match that criteria. And that is a little bit too many, so we will only like to have 100 for our downstream analysis. So we move this slider so that we get 100. If I move too many, so you will see that that's only one, then 3, 4, 5, 6, 7, 8. So here, get us to around 100. Okay, then those variables are presented here, but more conveniently, they're also presented here in a list that is already there. So what we have to do to store this result is to make a copy and call it result. And then we can add columns here, like Q value. And they are all very low. So at this point, we have a list of genes, 98, which have very low Q values for discriminating TL-AML1 group. So that's a strong statistical significance, and these are, in that sense, correct results. We can then visualize it. We can use a heat map, and let's order that, for instance, according to our leukemia subtype we would like to see. So we have the colors here, and maybe we would like to do hierarchical clustering on the genes here. Then we can go up in size here, so you can see better. Almost one step too much. So again, how easy it is to configure something. And if you would go back to the PCA plot, it's there, and the heat map here. And it looks good. Here are the upregulated genes, and here are the downregulated, and you see that that's an overall a good difference between those genes for that blue group, the TL-AML12, compared to the others. But if we start, don't continue, we are missing information. So now I will show you what the visualization can help you with. So if we would have applied hierarchical clustering on the samples 2, one thing pops up, and that is that there seems to be one green sample that is really close to the other blue ones. Don't know why, it's just how the data is constructed. And we would have picked that out in the PCA plot also directly, because it is so clear here. So if I would have done this the way we would do it, instead of starting with a table, you would actually just have done like this, and you get your same 98 genes if I hit the same settings. But all the time when you have done the statistical filtering, the results and the fact that the one green sample here is very close to the blue one, and maybe need some extra attention to find out why it seems to be very close or very similar to the TL-AML1 samples. Okay, now let's see. These were findings we believe in, and I would like to show you how you can compare those results with an external different data set. So I will download a GEO data set, Gene Expression Omnibus. You just have to write the session number, and then we will download it for you. And it will be soon be shown. So now I have two data sets open that are completely different. So they are not synchronized in that sense that you saw before, because they are really different. If we look at that data set, you will see that if you look at the identifier for the variables, you can recognize it as probe set IDs. So we need to manage that, and you can do that from the data tab. And if there is another annotation, in this case gene symbol, that you would like that to be the unique identifier, you can just change. And then you need to manage the fact that there are more probe sets matching to each gene symbol, so we can collapse the data for you. So now we get unique rows. So now I have gene symbols in both data sets. What I'm going to do now is that I'm going to take my result list, the 98 genes generated from the leukemia data set here, and see how they affect the second data set. And I do that by simply clicking that box. So now I use that result list as the base for both data sets. And as you can see, it seems to separate that new data set in two groups. Let's try to find out what that is. And in a similar way, we use the coloring and the visual inspection. And what we can see here is that we seem to filter out a lot of yellow samples, one purple, and 4 or 5 orange, at least 4 orange ones. Okay, let's look. The yellow ones are the TL-AML1s, according to that study. The purple one is TL-AML1 plus hyperdiploidy, and there are 4 others that also pops up into this group. So now I have actually shown you how you can use your results from a first study test, and within just a few minutes check it towards a second data set and really verify that that list of 98 genes are extremely capable of discriminating a subgroup like the TL-AML1s. And now I like that result then, so I will like to keep that, but I don't want this second data set with me, so I will discard that and instead create the log point for TL. Discriminating, something like that. Now I have those two log points that I can go back to later. So I will now change back to my slides, and to summarize the second demonstration, we have less than 10 minutes found new results and we have visualized them. We have verified the findings in the second data set. During the steps, we have, with the help of visualization, been able to have full control of what we did. And I also showed you how we could find that specific sample very easily with the help of the visualization. The benefits are obvious, and to build on them, visualization-driven analysis need to be implemented in the organization. To do that, the following areas need attention: deployment and customization. In Qlucore, we have native Mac and Windows solutions, and we have templates to control workflows and as a help to easily get you started. The templates are either developed by you in your organization, or you use a number of the standard templates that we ship with the program. The templates can be written in Python. A second important aspect of how to apply this to an organization is to have onboarding that is easy and fast. And the visualization always gives you that, and templates can make it very easy. And the fact that you can run it on platforms that you're used to is also a key on that. When you have been using the visualization-driven analysis for a while, you need to continue learning and enhancing. And we run a number of webinars and trainings, and the fact that teamwork is so easy with a visual approach, you can also learn from your peers. Which leads me to this section, which will be used then in the third demonstration on how the log points actually assist in this. But before that, let's have a look what more is included to support you and the team and the different tasks. So log points is one of the tools that we have to support you with. The team members, as you will have different tasks, and you will be working in different phases that change over time. Typically, you will be doing exploration and analysis. At some points, you might need a colleague to look at this, and then log point is an excellent way to share your analysis with a colleague. At some point in the analysis, you will move into formal reporting or need to do that, and there are many ways to support you with that in Qlucore. All plots, videos, and lists can be exported in standard formats and be used in any environment. You can also use the built-in PDF report to get a summary of everything you have done, including plots and references to statistical methods and so on. And in general, log points in the PDF report is very useful for keeping track of what you're doing. We believe that Qlucore will be one part of your workflow. You cannot do all steps and analysis required in one tool, and that is why we have a very generic approach to how data is imported. We are very flexible in that, and how data is exported. So we never lock you in. Instead, we try to facilitate import of data and export of data. Our template tool is one aspect of that. We also have an open API to R where you can add statistical tests, and we have scripts for converting data sets in R to Qlucore and back and so on. So making it easy for you is key for us. Before I would like to show you then the log point functionality and how. So now I'm here, I'm working with this data set, and then I realize I would like to check something that I did earlier. So I would just like to go back, and then I have just to select my log point and press restore, and then the program is restarted and everything is reloaded to that point where we were. I'm not sure you remember, but this was the result of the initial inspection that we did. We had found these groups, we have the variance filtering, and we have the networks. And then most importantly, when you have restored a log point, you can continue the analysis from that point. So it's not a screenshot, it's actually full restore of all settings in the program. So if I now would like to change something, let's say I would like to continue the analysis, I can just do it like that. So extremely convenient, and you can also send a log point to a colleague who have Qlucore installed, and they can then just get to the point where you were exactly in your analysis. The other thing that I mentioned was the PDF report. That is a convenient report to get a summary of what you have done. You can webinar. So you get some basic information here, the data set that you used, your own text, you can write as much as you want, then exactly the calculations that were applied to the data set. Then you get a list of the sample annotations that wasn't included. So details exactly on how you have treated the data set. Then you get plots, all open plots. Now we only had one, and with the legend here. And then comes a list of references and algorithms that are implemented in Qlucore, which means that it's always easy for you to go back and explain what you have done to someone. You don't have to think about that by yourself. You will have it here. So combining this information with these references, and you have a complete overview of what you have done. So, to summarize, for the individual user, it is possible to do better discoveries since he or she is in full control and can test more ideas than earlier. Work is fast and easy, and he or she is saving time. For the organization, the involvement of domain specialists is easier, and this leads to improved results and fewer mistakes. It is easy to drive and demand good teamwork. Clear questions from biologists to bioinformaticians save time and resources. So I have shown you a lot during this webinar, but more importantly is that you actually try it yourself. It's always good to believe in visualization and go and see for yourself. Thank you.
Brilliant. Thank you, Carl, for that interesting webinar.
Thank you.
So in the meantime, we have a couple of poll questions, as I mentioned earlier, and we'd just like to pass to the audience again. And next one up is the one I asked earlier about what type of data analysis do you believe will be the most important to you in the next 12 months? And the responses we had for that were gene expression, RNA sequencing, chip sequencing, single-cell RNA sequencing, proteomics, or other, and feel free to send any suggestions. And our final one was, will someone in your team try Qlucore Omics Explorer, yes or no, to get a feel if we're reaching the right people. Thank you all for participating in that. We'll move on to the Q&A. We've got a few questions coming through. So I have one for you first, Carl, which is, can I try by myself? And I'm guessing this refers to the demo that you're able to go on and look at.
Yes, you can absolutely do that. Just go to Qlucore.com and we have a free trial. It works for 10 days. It has full functionality, except with some limitations of exporting plots and results. So it's just to go there, register and download, and we will be happy to support you during the trial.
Brilliant. Thank you for that, Carl. I've got another one here. How easy is it to get started?
It is really easy. We have a nice suite of supporting information, both written and, of course, video-based. So you can get directly started doing the analysis, the type of analysis that I tried to show you here today. You can find discriminating variables. You can do gene set enrichment analysis integrated in the program, and visualize and explore in many ways.
Thank you. Our next question is it possible to integrate the program into a workflow?
Yes, and the solution to that is what we call templates. They are shipped with the program so that you get a number of pre-configured templates that assists you in doing standard operations, so that you can do it with even fewer mouse clicks than I showed you today. It is also possible for an organization to write their own templates, which means that they can get exactly the configuration of a workflow in Qlucore as they like. And then for the import and output, we take a very generic approach using standard file formats as much as possible. So you can get data in and you can get data out easily from the program. So yes, it is easy to integrate it into workflow.
Thank you. I've got one here about the timeline. So what makes the NGS module in Qlucore different with the conventional module? Is the difference just on the visualization part, or is there something else?
Okay, that's a good question, and I didn't show the NGS module. The NGS module is built up by several components. There are one, which is, you can say, a generic genome browser, but what is unique with that is it's coupled with dynamic filters on genomic entities in the same way as you saw me use the filters early on variance sliding and so on. So in the same way as I interacted with the program here on expression data, you can interact with it from NGS data. So that is really a unique flexibility and functionality that I think you should try out if you have NGS data.
Thank you for that. I've got a couple of more technical questions here about the actual some software issues. And the platform, is the platform available on Windows, Linux, or Mac computers?
It's available on Windows and on Mac.
Brilliant, thank you. And I've got a question about the input file format. So for RNA sequencing, what is the input file format that you have to use?
So there are several options depending on how much of the functionality you would like to use in the program. If you would like to use all built-in normalization, you would start with an aligned BAM file. If you already have done normalization, or you can also import it as a text file.
Thank you. And staying on the software, is there a one-time fee for the software? How much is the software?
Yeah, we license it for a yearly fee or annual fee, and then we include upgrades and updates, and so you're always having the latest version and latest functionality. As for the pricing, I recommend you to reach out to us, Qlucore.com, and the contact page, because it will be different if you're in academia or industry and how many users and so on. So reach out and we will be happy to give you a good price.
I've got another question on the software. Is it possible to analyze a whole transcriptome in a single go? And the question is alluding to, is there any limitation of data size with this software?
It's a good question, and it's a little bit tricky to answer. Yes, you can absolutely, but I will give it an answer in several steps here. You can absolutely analyze a whole transcript in a single go. I have been running, or we have been running it with hundreds of transcriptomes in parallel. Then on the other hand, of course, there are limitations in how much memory, how much hard disk, and so on, since the program is running locally on your machine. So yeah, but it's easy to sort out, and we can discuss in detail those type of questions if you reach out to us.
Well, thank you very much, Carl. I think that's pretty much all the time we're going to have for today. I'd like to thank you, Carl, for today's discussion and presentation. And again, for answering the questions, thank you to the audience for submitting them. Thank you for everyone joining us today and for the questions and answering the poll. I hope you found it a worthwhile session, and we hope we can follow up with a demo or an on-demand webinar to make sure we really get those demos across to you. If we didn't manage to get to your question today, we will follow up with you personally. If you have any more questions, again, please feel free to email me at editor@selectscience.net. You can also download a certificate of attendance in the Related Resources tab at the bottom right of your screen. And if you want to listen today again or invite a friend to listen, we'll be releasing that on-demand webinar copy shortly. Goodbye, and thank you once again for joining us.